Northeast India and their anti-diabetic properties. It's a fraction of my research work during my PhD program, and I consider it as a backbone of my study. As we all know, that Northeast is a home of various flora and faunas, and we have a different habitat and different agroclimatic condition from rest of the other states of India. Northeast people have a habit of consuming wild vegetables, and among these vegetables, Solanum family is also one of them. And also, not only vegetable, we consider them, some of the plants, we consider them as a traditional medicine we culturally use, and then it also has pharmacology properties, or some of them are also used as a ornamental horticulture. In addition to this, they also have the ability to grow under different strange and adverse conditions, and most of them are rich in nutritional conditions. So this study was done on 12 different plants of wild edible Solanum species, which was planted under the design of randomized block design with 3 replications, and the whole experiment was carried out in the Department of Vegetable Science, CSF, Pasighat, and then the biochemical process was done in Basic Science and Humanities laboratories, and also some pharmacological properties was also checked in Institute of Bioresources and Sustainable Development, Imphal. Coming to the methodology part, the morphology traits were recorded after adapting from the International Board of Plant Genetic and Resource Brinjal descriptor, and some biochemical parameters. Sulasudan was also checked using some research paper following their methods, and these antidiabetic properties were also checked in order to know their properties. So for antidiabetic properties, we have selected 2 enzyme assays. Both are in vitro. First one is alpha amylase inhibition, and the second is alpha glucosidase inhibition. So these are the list of species which I have selected from different regions of Northeast, and we have selected 3 genotypes of Solanum gylo and 2 genotypes of Solanum violacea, which we came to know that there are 2 types. And for further observation, we have sent this herbarium to Manipur University, and they have concluded that both are the same species. The one is spineless and one is the with spine. So the other species are, so these are the pictures which I have collected with their flower, fruit, and the cross section. The first one is Solanum ferox, which I collected from the Manipur, from the border town of Manipur in Myanmar, Tecnopole. Then the second one is Solanum nigrum, which I collected from the Pasighat. Second one is Solanum gylo, which I collected from Manipur. Then the fourth one is Solanum gylo with purple color, which I have collected from Manipur. The third one is Solanum gylo green, which I have collected from Arunachal, and the fourth one is Solanum acrocarpum, which I have collected from Manipur. Here is the plant of Solanum torbum and Solanum spiral, Solanum villosium, and Solanum mimosum. Solanum mimosum I have collected from the Mesipuma region of Nagaland. And these are the 2 species with different character, where we have seen that one is with spine and the other is without spine. So coming to the results of the morphological characters, we have divided into 2 qualitatives and quantitatives. Coming to the observation on qualitative traits, we have found that most of the species have upright plant growth habit with their strong leaf lobing, and most of the plants have the corolla color is white, and we have seen that most of the plants don't have fruit petiole prickles. And since these plants are consumed on their vegetative stage, we have found that the commercial maturity color, most of them are green, with some of are deep yellow, purple black, milk white, purple, and deep yellow. At their physiological ripeness, we have observed that plants have, most of them have deep yellow color at their physiological ripeness, and then other colors also we have observed like black, fire red, poppy red, fire red, light brown, and deep orange, as well as yellow orange. Coming to the quantitative traits of morphology, we have came to know that since it's a collection of different species and genotypes, we have come to know that among all these species and genotypes, Solanum tortum have the maximum plant height, and then minimum was from Solanum villosium. And then from the days to first flowering, since it's a combination, since all these plants have different growth habit, that is annual perennial, we have found out that Solanum violacum, which spineless, took more number of days for the first flowering, and less number was obtained from the Solanum nigrum. For the number of flowers per inflorescence, for all the genotypes, we have come to know that Solanum tortum has more number of flowers in per inflorescences. Coming to the number of days taken for fruit development, we have observed that Solanum ferox was taken for take maximum days for fruit development, and for fruit laying, we have known that Solanum mimosum has a maximum fruit length, whereas Solanum nigrum have minimum fruit length and minimum fruit breadth. So for the biochemical parameters, we have checked from the extract of the plant, where this is the process taken place for the extraction of fruit from the plants. Where the first one is the where the plants are cut into pieces, dried, powdered, and then macerated in 70% of methanol. Where then it was then second continuously for 48 hours, and then it was filtered. Then the extract was again kept in the rotary evaporator to get the crude extract, and the crude extract was again dried up in vacuum evaporator to get the crystal powder. So, since the plants are from Solanaceae, we have checked the solacodine content to relate with the antidiabetic properties. So in solacodine, this table and the picture shows about the results of the solacodine content, and where we have seen that among all the species, Solanum nigrum have the most solacodine content with a value of 3.66 milligram per gram. Then the minimum was found on Solanum gillopurple with a concentration of 1.34 milligram per gram. And for the antidiabetic assays, we have done two enzyme assays, that is alpha glucosidase inhibitory and then alpha amylase. The first table number 5 shows the activity of alpha glucosidase inhibitory. So here we have taken a drug for anti-diabetic, that is acarbose. We have compared with this drug at different concentrations starting from the 1 milligram per ml. So this shows the different concentration in 1 milligram per ml. It has the inhibitory effect of 96.01%. From the positive control, whereas from all raised samples which I have selected from my study, I came to know that Solanum nigrum, the fruits of Solanum nigrum, have the maximum inhibitory percentage, that is 76.4. Then the minimum was obtained from Solanum white with a value of 40.17%. We also have noticed that due to the in the decreasing of the concentration, the percentage of inhibitory were also decreased. In the second method, to reconfirm that it has the antidiabetic properties, we have also checked their alpha amylase inhibitory percentage. So here also we have taken the positive control of acarbose, and where we have seen that acarbose in 1 milligram per ml, it has the inhibitory effect of 84.95%, whereas from all the samples we have come to know the Solanum nigrum have inhibitory percentage of 69.48, which was then the minimum was from the Solanum gillo of greenberry, which is very less, 39.3%. Here the same like the alpha glucosidase, we came to know that the concentration, when the concentration decreases, the inhibitory percentage will also decreases. So to check their IC50, that is the inhibitory concentration of the half amount of the sample, we have calculated their IC50, where the IC50 denotes the concentration of sample required to inhibit 50% of the alpha glucosidase activity. Here we also take an acarbose, as it is an antidiabetic drug used to treat the diabetes mellitus of type 2. So here we came to know that when the positive control needs, that is acarbose, needs 0.015 milligram in per milligram, milligram, where our sample result we got it was 0.163. Yes, the acarbose result was very much lower from our sample, but but our sample from Solana nigrum is quite comparable with the acarbose from the positive control. And the minimum was found from the Solanum green, which have a very maximum IC50, that is 1.25 milligram. Next is IC50 of alpha amylase inhibitory from the samples. Here we have seen that when the positive control acarbose have a value of 0.019 milligram, our sample Solana nigrum have the IC50 of 0.428, which was followed by the Solanum macrocarbon 0.09. Coming to the summary and conclusion, I would like to conclude that all this difference in morphology is due to the baffling range of growth forms such as herb, shrubs, and annual. And coming to three genotypes of Solanum gilo, we have came to know that most of the morphological traits of Solanum gilo of green berry and white were most similar, whereas the Solanum gilo of purple have different. And then the difference is in this Solanum violacum collected from Manipur and Arunachal have similar morphological characters except the presence of spines. And it also came to know that from this little knowledge, we know that this is under which we have been practicing from before, that these plants have some anti-diabetic properties. With this little knowledge, it has been proven scientifically that these plants can be eaten by the patients of diabetic. And coming to the future prospects, I would like to suggest that with this data, it aroused interest in the cultivation, and we have to give awareness to our local people. And then we need further research to explicit the compound responsible for this. And it lays down a great scope for the farmers in cultivation of this group of Solanum in northeast region of India. Thank you.